Hello and welcome to this Caroline Cymru biology revision session in which we are going to cover the immune response and the role of B and T lymphocytes, part of the A2 specification, the 4.6 unit. So first of all, let's get a little bit of an overview of what we're going to cover today. So when we think about defense against infection, really what we're can do is divide those up into a physical defense, a chemical defense, a localized defense, and then sort of a cellular defense. So let's look at each of those in turn. So when we're talking about physical defense, we're looking at physical barriers which actually prevent bacteria or pathogens from entering our body. So one of those is the skin. The skin is a natural barrier itself. It also has a pH of the skin is just a little bit lower than neutral, and it's a pH which prevents the growth of these pathogenic bacteria. It forms a protective barrier, like I said, as a physical barrier that stops bacteria or pathogens, I should say, rather than just referring to bacteria. I should say pathogens, because that's bacteria, viruses, or a pathogen is any microorganism which causes disease. So commonly we think of bacteria, but obviously viruses are also equally relevant, and really the correct term to be referring to here is pathogens. And also on the surface of the skin, you have what's referred to as skin flora, and those are what might be referred to as friendly bacteria, or certainly non-pathogenic bacteria, which thrive on the skin and outcompete the pathogenic bacteria. So there's an argument there that really you don't want to be removing those the skin flora, those non-pathogenic bacteria, because they will outcompete the pathogenic bacteria. Elsewhere, what other physical barrier do we have? Well, just the principle that blood clots, the platelets in the blood are there ready and waiting for when you do get a wound, that it clots very quickly. And again, that prevents a physical barrier. That scab forms a physical barrier to entry of pathogens. Then if we go into chemical, so there's a few examples here. So the first one is your tears. They contain an antibacterial enzyme. It's called lysozyme. And what that does is it destroys the cell walls of bacteria, and so destroys them. You're also probably familiar with the idea that we have stomach acid, and stomach acid then, whilst we know that pathogens are going to enter our body on our food, we want a defense against them, because we're sort of giving them a pathway into our body. We want a first line of defense for those that are given that access, and that's the stomach acid, which will kill most pathogens that come in on food. And finally, you have mucous membranes which contain these cilia. So what we mean by that is, you'd be familiar with this from Unit 1. So things like the trachea, the windpipe, the cells which line the trachea are ciliated, meaning they have these little cilia, these hair-like structures which move, which beat sort of in a wave-like fashion. And the mucus that's produced by these cells lines the trachea, and so any dust, microorganisms, including pathogens, if they enter the body, they'll often get stuck in this mucus, and then the cilia move that mucus back up to the mouth, and then it can either be swallowed or gotten rid of that way. So they are pathogens trapped by mucus, and then the cilia move the mucus up the trachea. Then we have a localized defense. So we're talking here now about, if you were to cut yourself, that is an entry of where bacteria can get in, and whilst blood can clot quickly, there still is a pathway for the bacteria to get in. So what do we do there? Well, first of all, we've got phagocytosis. So phagocytosis is carried out by certain white blood cells called phagocytes, which engulf pathogens, engulf them, if you like. You can think of engulf, you think of it like eating, but it's not eating, it's engulfing and destroying them directly. You also get inflammation, and when an area which has been damaged, if you've got a cut, when it becomes inflamed, what that does is it restricts or confines then that infection to the area which has been damaged. Okay, so the inflammation is a good thing. And you also have some local blood vessels which dilate. We call it vasodilation from GCSE, and that improves transport to the area. So actually what tends to happen is when the surface of the skin is damaged, what we really want is white blood cells, these phagocytes, to get there, to come there, and what happens, those phagocytes which are there will not only will they engulf bacteria, they'll release chemicals which attract other phagocytes to the area, and they're brought there, not least in the blood. However, today what we're going to focus on more than anything else, we'll have a little bit of a look at those other things, but is the two types of cellular response. So this is the one where there's a lot of detail, and those cellular responses involve the humoral response, which involves B lymphocytes, and then the cell-mediated response, which involves T lymphocytes. Okay, so we'll be having a look at those a little bit later. So let's just have a look at the type of question you can get. I'm going to try my best here to also give you a little bit of help with how you go about doing past paper questions, and more importantly for me, learning from past paper questions. So this question here shows a skin wound, and it asks you to describe 2 ways in which healthy, undamaged skin, so not really relevant to the picture, protects the body against invading pathogens. Well, 2 that I've chosen to add on here to say that the pH prevents the growth of pathogenic bacteria. I mentioned that already, and the skin flora outcompetes pathogenic bacteria. So those are the answers I've put down. Let's check against the mark scheme. So here it says that let's see what I've put here. So my first point of saying the pH prevents the growth of pathogens. You can see here that I've put a tick on, and corresponding to the skin pH at the bottom, and then the second point there that's been highlighted, the natural skin flora prevents growth of pathogens. Yeah, that's what I've got for that third point there. Okay. The other point mentioned the idea of a natural barrier to prevent the entry of pathogens. Well, I haven't said that. And I would just look at, there's a couple of underlined words, which means you have to say those words. I think when I mentioned this previously, I said it being a barrier, but I didn't add on this idea of preventing entry of pathogens. So I'm sort of learning from this that this is something I need to do in the future. If I was going to talk about a barrier, that I should be saying prevents the entry of pathogens. With the one underneath there, now it says, I'll just take that away a moment, it says, damage to the body's tissues triggers a localized defense response called inflammation. Using the above diagram, written in bold, and your own knowledge, describe 3 ways in which the body protects itself from bacterial invasion at the site of a wound. So these are the answers I put down here. So I'm saying phagocytosis occurs, so and I've given some detail. What I mean by that, white blood cells engulf bacteria. I've said local blood vessels dilate to aid transport to the area, and I've also said that platelets help blood to clot quickly. So let's check now against the mark scheme, and we'll do the same thing again. Okay, so there's the first one, phagocytosis, and that's that I've said. So I'm giving myself a mark there. The local blood vessels dilate. Yep, I've said that there, with increasing diameter or vasodilation to increase transport. So I've said that. And then the third one, look, it says blood clotting. And then it says to seal the wound. And the seal the wound is underlined, and I haven't said that. So for me, that third point doesn't get the mark. I've said it clots, fine. But I'm adding now to what I've written on. You can see that I've added the words to seal wound, and I've underlined it. And that really, and I said that this is about me trying to help you to learn from past paper questions. That now is the most important thing that I've put down on that page, because that's now saying this is what you don't know. So this is what I need to include next time I talk about blood clotting. Okay, they've got it in the mark scheme underlined. And so I'm going to make sure I say that next time. You can see they've also got new cells replacing damaged tissue. I don't think, so I'm making a judgment there that I don't think that's something I want to recall for next time, because I'm thinking, I haven't seen that before. If I started to see that in lots of questions or lots of mark schemes, then I'd start to think, right, I need to know that because this comes up a lot, but I haven't seen it before, so I'm happy to leave that out. So let's now focus then on this cellular response. So we've looked at that first line of defense, and we're now the cellular response. So this is what happens once a pathogen properly enters the body, and how do we go about fighting it off. So the cellular response is divided in two. You can have the humoral response, which involves B lymphocytes, a special type of white blood cell, or cell-mediated response, which involves T lymphocytes. So let's deal with the humoral response first of all. So the first thing that happens in the sequence of the humoral response is you have these foreign antigens. Now, an antigen, the word antigen comes from the word antibody generating. So an antigen is a some sort of molecule marker on the surface of a any cell which does not belong to you, really, that you recognize as being foreign. Now that can be, most likely it'll be a protein on the surface of the pathogen that then gets detected by an antigen binding site on a particular B lymphocyte. So in your spleen and in your lymph nodes, you have a huge number of B lymphocytes, each of which is subtly different, each of which has on its surface a binding site which is unique. It's unique in the sense that it is unique. It is no other B lymphocyte would have an antigen binding site like that, and also that it will be unique to a particular antigen. So each foreign antigen, what we know is that for every foreign antigen that enters our body, there will be a B lymphocyte which is closely it is specific to it, okay, that will be detected by it. Not all, so all B lymphocytes are unique, and only one will be specific to a particular foreign antigen that enters the body, okay, and all that happens in the spleen and the lymph nodes. So when that happens, once that antigen has binded with the antigen binding site on its specific corresponding B lymphocyte, we get the next stage, which is called clonal selection. And the clonal selection is where that particular selected B lymphocyte, we say it proliferates. And it's an important word to use there. We can say divides by mitosis, but the key word to use is proliferates, to produce lots and lots and lots of copies of itself. It's like we're saying this B lymphocyte is the one that we now need to take action, because it is the one which is specific to the foreign antigen which has entered the body at this time. So that particular B lymphocyte, we need to make more copies of it, and it proliferates, divides by mitosis. And it produces then two populations. So the first population it produces are these things called plasma cells. Now, plasma cells don't stick around too long, so we say they're short-lived. Their job is to release antibodies. Now, the antibodies will be specific to the antigen, the foreign antigen, because the antigen binding site on the B lymphocyte was specific to the antigen. So really, these antibodies are much like the antigen binding site, but they are not attached to the B lymphocyte. They are free to move and enter circulation and find other antigens. The correct term for antibodies is globulins, but it's okay to use the words antibodies. The key thing here is that they are specific to the antigen, or specific, therefore, to the pathogen. Now, if you look there at my diagram of antigen binding site, you see there are 4 polypeptide chains in an antibody. You've got 2 red ones. I mean, they're not red, but just trying to show the difference here, and 2 black ones. And they're all joined together by disulfide bonds and perhaps other bonds too. The red ones are smaller, and so they're referred to as the light chains, and the black ones are longer, and they're referred to as the heavy chains. And at the end of each, so at this area here, not showing that particularly well on this, there we are. At this area here, that's the bit that's variable. That's the bit that will actually bind to the antigen. And you can see on this particular antibody, there are 2 antigen binding sites. Okay, next thing. So what happens to those antibodies? They've been released. Millions, billions of these antibodies have been released. And those antibodies then are enter circulation, and they will move around the body, and they will find other antigens and bind to them. We say they bind to them in a complementary fashion because it's a specific arrangement or a specific match between the antigen and the antibody. So they bind to each other just like an enzyme and a substrate bind, okay? And remembering that they're specific. So antibodies bind to the complementary antigen and they form what's called an antigen antibody complex. So the diagram we're seeing down here now is an antigen antibody complex. That happens repeatedly. So you notice there's two binding sites now on an antibody. That means one antibody can bind to two antigens. So I can show it down here that we've got one antigen binding site has bonded to an antigen there, and another one has bonded there. And so we get what's called agglutination. Okay, that is all of the antibodies and antigens sort of clumped together, and we refer to that as agglutination. And then the next stage is when we get all these pathogens that have been clumped together. At that point, we get engulfment by phagocytosis. So large white blood cells will just engulf the whole lot, this whole complex, this whole sort of mass structure of agglutinated antigens or agglutinated pathogens. So if you go back to that localized defense, where you got phagocytosis by white blood cells, but in that case it's quite slow, so it's quick to start. As soon as you get a cut, that those white blood cells will carry out phagocytosis, but they will do it on individual antigens, individual pathogens. It's far better to do it this way, whereby we get agglutination of the antigen and then the white blood cells can engulf the whole lot. But this takes time to set up. That takes a few days, maybe even more than a week, for clonal selection to take place and for the B cells, B lymphocytes, to proliferate, to produce copies and form all these plasma cells and go and release these antibodies. It takes time for that to happen. But once those antibodies are released, what happens then is very quick and very effective. It just takes a bit of time. So I said there's two populations. The other population that's produced are called memory cells. So these are not short-lived. These remain in circulation for a long period of time. And their job is to detect that same specific antigen if it ever enters the body in the future. And if it does, the memory cells divide into two populations. They divide into plasma cells, but do so much quicker than when it was going by that original process of clonal selection of that single B lymphocyte that was in the spleen or lymph nodes. Now, because these memory cells are already in circulation, first of all, it's easier for them to be found. And secondly, they're activated. That is, they form these plasma cells much quicker. They proliferate much quicker. So they form plasma cells, and we get the same process happening if that antigen enters the body. And we also get memory cells to sort of reinforce this idea that we need long-lived resistance against future entry of that antigen. Usually, it's so quick that that when those memory cells divide, it's so quick that we don't even recognize any symptoms. We don't get any symptoms from the pathogen. So the first time we will get symptoms because it takes a long time for clonal selection to take place and the B lymphocytes to proliferate. But after, so it takes a long time for that to happen, a week or two. So we'll get symptoms. But if the same pathogen enters the body again with the same antigens, it's much, much quicker and we don't get symptoms. So we'll say, well, I've only had chickenpox once. It might be that you've had chickenpox more than once in your body, but you produce such a quick immune response the second time that you never knew you had it. Right, let's have a little check against these. So I won't spend long on this. You are welcome to pause at this stage and have a go at regurgitating these words, but I'll do it with us straight away. So number one is an antigen. So foreign antigen detected by an antigen binding site on a corresponding B lymphocyte. Then we get number three, clonal selection, in which case a selected B lymphocyte proliferates. Remember I said that was a key word. And then we form two populations, one of which are the memory cells, which detect the antigen if it enters the body in the future. Over to number 7 there, am I right? Yeah, so plasma cells, they release the antibodies, number 8, specific to the antigen. The antibodies bind to the complementary antigen, forming 9. Have I missed one there? I've missed 9, sorry. An antigen-antibody complex. And I'll just go back to here to do one at a time. Number 10 is we get agglutination of the antigen. 11 is engulfment by phagocytosis. And 12, this key idea that it's quicker the second time round. Here's another little activity you can have a go at. Same thing with in terms of filling in the blanks. You can pause, but I'll go through it now. So natural barriers. So look at the question here. Describe ways in which the entry of pathogens into the body is prevented. Also describe how the human body defends itself against infection with reference to the humoral immune response. So prevent entry of pathogens. We talked about natural barriers to infection include the skin. We talked about the skin flora and the pH. Ciliated mucous membranes. Remember the cilia on those cells. And lysozyme in tears and stomach acid. So that's really our natural barriers, the first bit. Now let's talk about the humoral response. It's the presence of antigens on the pathogen which stimulate B lymphocytes to proliferate. In doing so, they form 2 subpopulations, plasma cells and memory cells. Plasma cells produce antibodies. These are Y-shaped molecules consisting of 4 polypeptide chains which are carried around in the body. They bind to the complementary antigen and form an antigen-antibody complex. And these all bind together in a process called agglutination. And they undergo phagocytosis by macrophages. So I didn't mention the term macrophages before, but macrophages are a particular type of white blood cell which carries out phagocytosis. You could also refer to them as phagocytes, and that ties in with the process they carry out. The memory cells, or the other subpopulation, remain in the bloodstream, give long-lasting immunity, meaning that if the antigen enters the body in the future, antibodies would be produced more quickly. And that quicker idea is something we see a lot in questions. So let's look at the cell-mediated response. Okay, so this is the last thing we'll look at now. Lots of similarities here, but there's a little bit more going on. So the first thing happened, I've rewritten it in the same way. A foreign antigen is detected by an antigen binding site on a corresponding, now it's a T lymphocyte, and the location of this is in the thymus gland, okay, sort of in the upper in your upper body. That then undergoes, that T lymphocyte, that specific T lymphocyte undergoes clonal selection. Same thing, selected T lymphocyte proliferates, means divides by mitosis, but now we produce 4 populations. So there's a bit more going on here. So the first subpopulation, or first population, is T killer cells. They have other names, and it's not a particular helpful thing, but they got multiple names. We have T killer cells, or effector cells, or cytotoxic T cells. Now, the only reason I write all 3 is because it's possible that they may be referred to as other things, and you need to know they're referring to T killer cells there. And their job is to cause lysis of infected cells. Lysis means splitting, so it just sort of directly they directly kill any infected cells, that cells that have been infected. This is probably more relevant with a virus, as you get infected. Then we get helper T cells. Okay, now helper T cells do a lot. So this is the second type now, the helper T cells, second type of or second population of T lymphocytes. So the first thing they do is they cooperate with the B cells, the B lymphocytes, to initiate this antibody response. So we know about the humoral response, but they enhance that, and we'll look at that in a moment as to how they do that. They also release cytokines. Now, what cytokines do, they enhance that or stimulate that clonal expansion of B lymphocytes. They also stimulate B lymphocytes to make antibodies. So really, if I take that back a bit, I said that helper T cells cooperate with the B cells to initiate the antibody response. And then I said they release cytokines. And then I said what the cytokines do, they stimulate. Well, these 2 things I've now said, they stimulate clonal expansion of the B lymphocytes, and they stimulate the B lymphocytes to make antibodies. They enhance that. That really explains that first point, they cooperate with B lymphocytes. So that's why I've shaded that out on this slide, because if I just take that back a little bit, there's nothing wrong with that. They cooperate with the B lymphocytes to initiate their antibody response. And now I say, okay, let's give detail on that. Okay, well, what they do is they release cytokines. And what the cytokines do, these chemicals, these cytokine chemicals, is they cause clonal expansion of or the proliferation of the B lymphocytes, and they encourage them to make antibodies, which then explains what we mean by they cooperate with B cells for this antibody response. So I've given details to what happens there. It's not the only thing they do. They also stimulate those phagocytic cells, those phagocytes, for example, macrophages, to engulf pathogens. So they have a key role in multiple parts of the immune response. We also have third population of T lymphocytes are memory cells. Well, these work in the same way as the B lymphocyte memory cells. They just remain dormant, remain in circulation for when that antigen enters the host in the future. And the same thing applies. They then proliferate into 4 populations, like we're seeing here. And finally, we have suppressor cells. So suppressor cells, their job is to suppress the immune system, or to switch off the immune system once that antigen is no longer inside the body. So you can sometimes have diseases where these suppressor cells don't work, and you've got sort of like a hyperactive immune system. So these are really important for just calming things down afterwards. Right, so let's have a look at the same ideas I showed previously. So again, you're welcome to pause this and have a go yourselves, but I'll go through them now. So the foreign, what should we have there, is the foreign antigen is detected by an antigen binding site on a specific T lymphocyte. Remembering you've got a huge variety of T lymphocytes in the thymus gland, only one of which will be specific to any antigen which enters the body. When that one has been selected, it undergoes clonal selection. To clone something means to produce lots of copies, isn't it? So we're selecting that T lymphocyte for cloning. So it proliferates, number 4. That is, it divides by mitosis, which we said previously, and we get 4 populations. So number 5 is T killer cells, and those cause the direct, well, killing, bad word to use, but lysis of infected cells. Helper T cells. The helper T cells, they release cytokines. So you notice I haven't said that they help with the humoral response, the B lymphocytes. I've now just explained it. So they release cytokines which stimulate the proliferation of the B lymphocytes and stimulate B lymphocytes to make antibodies. And they also stimulate the phagocytic cells, such as macrophages, to engulf a pathogen. Number 11 here, what else do we produce? It's memory cells. And number 12 is suppressor cells. Memory cells, they just stay there, waiting for the antigen to enter the body again. Suppressor cells, their job is to switch it off, the immune system off, when the antigen isn't there anymore. So you see there's a little bit more there with the T lymphocytes. There's 4 populations now, and memory cells is the same, but there's a little bit more going on here. You see why we have to do the humoral response first, before we go into the cell-mediated response, because the cell-mediated makes reference to the humoral response. The one other thing I'd say about the immune system topic actually is that different textbooks will refer to different aspects or focus on different aspects of the humoral or cell-mediated response depending upon the author. So the useful thing about what you're seeing right now is that what I'm presenting here, corresponds to the WJC's preferred version. I don't know if that's the right way of putting that, but the things that the WJC specification wants you to know, and the terms the WJC specification wants you to know as well. You can find a whole host of different terms for the same things in textbooks, but know that this is the version that you need. And if you were to go on YouTube or to just look up any random textbook, you will see things that I'm not going to say contradict what I've said here, but will give other sides to it. So it's best to try and stick with the version I'm presenting here. Right, okay, here's a question. Describe how the human body defends itself against infection with reference to the cell-mediated immune response. Same idea. You can pause if you wish. Let's have a look at the answers. The cell-mediated response involves T lymphocytes. They are activated in the thymus gland. Okay, that's or proliferation, or they proliferate from the thymus gland, or they're selected in the thymus gland. Here, each T lymphocyte has receptors which recognize and fit with a specific antigen. And then that T lymphocyte proliferates. We've said that before, divides by mitosis, producing 4 subpopulations. T killer cells, which kill infected cells, T helper cells, which activate B lymphocytes to initiate an antibody response, suppressor cells, which switch off the immune system, and memory cells. Just on that one slide there, I would say is the minimum of what you need to know. There's lots of key terms that are included on there. If I can try and show you on here, I mean, what biological terms have we got? Cell mediated, T lymphocytes, the idea of them being activated, the location in the thymus gland, the idea they have receptors which recognize a specific antigen, and then the idea of proliferation into 4 populations. So the T killer cells, and what they do, well, perhaps rather obviously they kill. The T helper cells, and then we're linked to activating the B lymphocytes, and they initiate the antibody response. Suppressor cells, which switch off the immune system, and memory cells, and the idea that they quickly proliferate in the future. Now, on that slide there that I'm showing you is, I would class as the minimum. You've got all the information there. There's not a huge amount of detail. There's no reference, say, to cytokines, but what's on there is a lot of detail that will put you in good stead for your exam. Okay, let's have a look at a question. Okay, so this won't take too long. It's quite a straightforward question here. I'll try and do the same thing again, how I can sort of advise or help with learning from questions. So remember, when you do past paper questions, they're an excellent way of learning, but importantly, it's not about doing them. No one cares about, and you shouldn't care about how well you're doing them when you're using them for revision. The only thing you should be interested in is learning from them. Because really your job is to produce your best responses in the real exam. So do past paper questions, absolutely, but learn from them. Look to learn from them. Don't look to do them. Don't care about you getting a high mark or anything. The key thing is that you learn from them. So there we are. T lymphocytes and B lymphocytes produce different types of immune response as shown on the diagrams below. And it says complete the table to give the name of the responses shown in type 1 and type 2 above and name the lymphocytes responsible for each response. Now, this is a 4-mark question, which I think is an absolute gift. Your job is just to identify whether it's cell-mediated or humoral and know which is which. So I put the answers on here. Type 1 is humoral. How do I know? It's because it's producing memory cells and plasma cells. The memory cells isn't a giveaway, is it? But the plasma cells is. And I know that humoral involves the B lymphocytes. And type 2 then is cell mediated. Well, it's the other one, so I know I have to say that. What's the giveaway here? It's got killer cells, suppressor cells. It doesn't mention T helper cells here, but it can only be the cell mediated response, and that's T lymphocytes. So there's the mark scheme, very straightforward. I'm giving myself 4 marks there. I'm not learning anything from it because it's frankly too easy a question. But it says, following exposure to the foreign antigens, the lymphocytes responsible for each type of immune response differentiate. Describe the functions of the memory cells and plasma cells produced in the immune response type 1. So it's referring to just the humoral response. So this is the answer I put down here. So I'm saying the memory cells remain dormant in circulation. When the antigen enters the body in the future, memory cells recognize and antibodies are produced quicker. Okay, and I think that's a decent answer. I think I'm going to pick up 2 marks there. Here's the mark scheme. So let's see what I've got. Well, the idea that they recognize the antigen, I've given myself a tick next to that. The idea that the immune response is faster, or what I've said is the antibodies are produced quicker. Well, the immune response is producing antibodies, so I'm content to give myself a mark there. They mention here about a reduced a latent period, and that's something that comes up a little bit later on when you talk about, I suppose, vaccination and immunity. But I've made a note of it here because I'm looking at it. I'm thinking, so with my experience and thinking, knowing what comes up in other questions, I think, do you know what? It's a good thing to include the latent period. We haven't talked about it yet because we don't really cover that today. But the point I want to make here is not that you should know what the latent period is, but to look at that, the answer, the mark scheme, and think, is there anything else that I think that would be worth me including in my answer were I to do this in the future? And you notice the color I choose for that. I've got that in bright red. I've got my ticks in green. I'm not really interested in my ticks. That's information I know already. As a student, I'm interested in what I didn't know. So even though I got the 2 marks, that doesn't mean that I should forget about everything else. What else was on there? Is there anything else on that mark scheme that I might go, oh, that's a good point. I should use that next time. And the next question, what about the plasma cells? Well, the plasma cells, I've said they produce, and this I think was trickier, because what do they do? I want to say they produce antibodies. Okay, so, but I'm not sure what else my other marks are coming from. So they produce antibodies that are specific to the antigen. And then I've gone on to talk about the antibodies. These bind to the antigen and cause agglutination of the antigen before being engulfed by phagocytosis. But I'm not sure about that because I was looking for that 2 marks. I'm thinking, where are my 2 marks coming from? So I've written enough, and what have I said there? I've talked about the antibodies being produced. I said they're specific to the antigen. I say they bind to it and cause agglutination. I could have said form an antigen antibody complex on there as well. But I also talked about phagocytosis. There's lots of sort of ways in which I could pick up marks. But I'm not really, the second sentence is answering the question of what plasma cells do. Here's the mark scheme. So the mark scheme says that plasma cells, this is taken directly from the mark scheme, they synthesize or produce specific antibodies. Well, I'm happy that I've said that. They produce specific antibodies. I said antibodies specific to the antigen. And the second mark on there is for saying that they release those antibodies, okay, into the plasma or into the blood. They release those antibodies. Well, I haven't said that. I think that's quite a nasty mark scheme. But hey, I didn't get the marks. I just need to make sure, and it's strictly speaking, it's true, isn't it? Inside the plasma cell, those antibodies will be produced, fine, but they then got to be released or secreted. So I haven't given a thorough enough answer. So I'll just bear that in mind. Yeah, I could have been clearer on that. So I've made a note of it in red there. I should be thinking of that next time. So there's not just so in this session, I haven't just talked about then the facts that you need to know. I haven't just asked you to do a little bit yourself and sort of those fill in the blanks exercises. You could even have done these past paper questions, but I'm really also trying to encourage you to think about your the way you answer past paper questions and how you can learn from them and appreciate. And the most important thing is to identify the things you don't know, because that's what you want to take forward into your next question and ultimately your final exam. Okay, so I hope you found that useful. You can find more videos at the Callum Cymru website relating to biology and other subjects as well. But for now, that's it from me.